that's that info. And so today we're going to continue thinking about cellular innate immunity. And I will admit, with the stuff I'm talking about today and the stuff I'm talking about on Monday, this is stuff that is very much related to my research. It's very near and dear to my heart. I can geek out hard if you guys have questions. So we started talking about the general steps of inflammation last time. And if you recall, we started out with this skin site that was living happily ever after. It had a tissue cell, which was a macrophage, hanging out there. And then we got a wound that allowed some bacteria in. And we talked a little bit about that macrophage sensing that those bacteria that have come in and starting to produce these new products, the inflammatory cytokines. So we saw this last time. I told you about pattern recognition receptors, which are some receptors that those macrophages have that allow them to detect the bacteria. And that's one of the areas that I'm going to get more into later on. That might be a Monday thing. But now we want to see kind of the rest of the steps of what happens in this process. Largely starting from what happens with these inflammatory cytokines. What do these inflammatory cytokines do? You can also see that this the title of this slide I have listed as what do inflammatory cytokines and chemokines do? And so I just want to define the term chemokine, just like I defined the term cytokine, because some of the slides I have mentioned chemokine, some do not. Chemokine is basically like a shortening of a longer term. The idea of chemokine is that it is a chemotactic cytokine. So basically we're taking the word chemotactic and we're taking the word cytokine and we mush them together and we make chemokine. Chemotactic or chemotaxis means movement in response to chemicals. So like movement towards or away from a chemical. So the idea with a chemokine is that it's a cytokine that makes other cells move. Like it's in fact, there's this really great cartoonist who does immunology cartoons. Sometimes you will see them that I'll randomly put them in. And if I had been thinking well, I could put one of his cartoons in right here, where he shows a cell making some chemokines and the cell says, my chemokines bring all the boys to the yard. Like that's the idea, is that chemokines are really sort of making other cells move. And so there are also some chemokines being made by this macrophage. But as I move into sort of some of these details of inflammatory cytokines and chemokines, I want to sort of think about one other aspect of inflammation. So inflammation was actually first described a lot of years ago. The stuff I'm going to mention to you has to do with how the Romans talked about inflammation. And the reason why I mention that particularly is that the Romans did not have the Bio 250 prereq for this class, yet they could still say a fair number of things about inflammation. Because while they didn't have the Bio 250 prereq, they did have bodies that they could make observations about. When they described inflammation, they talked about 4 things happening to the body. They are now known as the 4 cardinal signs of inflammation. I'm sure you all know them from your life experience. So if you have inflammation, what are the 4 things that tend to happen? You don't have to name all 4. You can just name one. Yeah, Brenna. Redness. The area gets red. The Romans called it rubor, but it's redness. What else? Yep, Shelby. Swelling. Definitely. The Romans called it tumor. What else? Yep, David. Heat. So the area gets warm. Romans called it calor. Yes. Pain is the fourth one. Romans called it dolor. And so they talked about the 4 cardinal signs of inflammation: redness, heat, pain, and swelling. All 4 of those really come back to the actions of the inflammatory cytokine. So the inflammatory cytokine, in a lot of ways, is actually eventually going to lead to each of those observable body changes. You will see places pretty easily for what goes on with the redness, the heat, and the swelling. Although some of the heat stuff I'm not going to go into total detail about. I'm not going to say a ton about the pain aspect of it. I actually did not know back in the day that how much we did or did not know about the details of this. It turns out that at least when I was in graduate school, we didn't know a ton about how that exactly the inflammatory pain happened. And I say that because it turns out that currently the world's expert was one of my classmates from grad school who does all, and so I'm kind of I sometimes will look at work that Isaac did and be like, guess we didn't know that before. His lab basically works on why do immune things itch. And who knew? We didn't know that. But the moral of the story, which again, I'm not going to say much more about any of this, is that really the inflammatory cytokines are acting on local nerves. And since we're not going to talk about the whole nervous system, I'm just going to say the cytokines did it. This image also shows some other things that the cytokines are doing. I'm going to come back to this image in a second. But here you can see some of the kind of classic inflammatory cytokines that a macrophage makes, along with their specific actions. So you can notice when I think about sort of the classic inflammatory cytokines, I often think about TNF-alpha, IL-6, and another one that's not listed here. Oh, it's up there, called IL-1 beta. But you can also see we've got some other cytokines and chemokines listed here. Like I said, in my mind, it's TNF-alpha, IL-6, IL-1 beta. And this shows you some of the things that these cytokines do. I'm not going to ever ask you, tell me which function is TNF's function as opposed to IL-6's function. We can sort of group all of these together. But what you can notice is that one of the things that happens is that we see some changes to the blood vessel. And that's listed here as something that's happening as a result of TNF. And so I'm going to move to another slide and then I'm going to come back to this. So here's my blood vessel before the cytokines. And here's my blood vessel after the cytokine has actually acted on these cells. So this little triangle cytokine is acting on these cells of the blood vessel wall. What do you observe is happening to that blood vessel? Yep. It's dilating. It got way bigger. So we have dilation, vasodilation of that blood vessel as one major change. And there's a second major change that's happening as well. Yeah. It also gets more permeable. So you can see that there are bigger gaps between these cells. So we've also got increases in vascular permeability. What do you think can happen, or what do you think happens to this area of the skin when the blood vessel gets bigger. Like, why is that useful, I guess, first of all, in terms of immunologically? Why is it useful for the blood vessel to dilate? Yeah. Generating more blood flow, you can kind of imagine it. We make a bigger highway. More blood proteins, more complement can come, more immune cells can come because we got a bigger highway for them to come on. And so we can get much more blood flow coming to that location. Why is it useful that this blood vessel wall gets leaky or increases its permeability? Yep, Brenna. So it can recruit more macrophages or other cells. It doesn't have to be a macrophage. In fact, you'll see later today, it's often a neutrophil at this point. So it can recruit them, but also so they can actually get out of the vessel easier. If these cells need to come in and help out in this area, having a leaky blood vessel wall makes it easier for them to go into that location. You can also imagine it's easier for things like complement proteins that might have been in the blood to go into that location when the blood vessel wall is leaky. What's going to happen to that spot on your skin if the blood vessel gets leaky and gets bigger? It's going to swell. Why is it going to swell? So you're going to get more fluid in the area and you're going to get swelling. So in fact, that already shows you one of those ways that the cytokine leads to one of the cardinal signs of inflammation. What else might happen to that area? Yeah. Gets red. Why is it going to get red? Because you have this influx of blood proteins and blood cells. And so the redness is going to be partially coming in, again, from the same exact influx. Anything else that might be partially related here? Yeah. There maybe could be some pain. That is definitely a possibility from kind of like the area just being so packed, potentially. There's one other thing that I would think about. Yeah. Maybe heat. If you think about it, the blood is usually a bit warmer than a lot of the extremities. And so as you get that nice warm blood going into places, that's also going to partially increase the temperature. And so just from that blood vessel change, you can imagine ways that all of these cardinal signs of inflammation are going to happen. As I am telling you this, there's one other thing I want to say. I'm just noticing that this slide is not in the order that I'd want it to be in, but that's okay. And so this also indicates one other piece of information about this inflammation, which is that this inflammation that's happening is local. So if we imagine a wound on the skin of the hand, you can see this hand gets red. And what your textbook is trying to tell us here is that we're seeing those changes in the blood vessel just there in the hand. It's not like every blood vessel in the entire body has gotten bigger, or every blood vessel in the entire body has gotten leaky. It's just the ones near that site that are getting the cytokines from that macrophage. So the rest of the body, the heart, the liver, the spleen, the kidneys, all the rest of the body does not have this inflammation going on. It's just in this one small local location where we're going to see changes to the blood vessel. So yeah, it's sort of, just these blood vessel wall cells that are actually, interacting with the cytokine. It's not the ones in like the foot or something. So that's one big thing that's going to happen because of those cytokines and chemokines. We also, if I go over to these two, are going to have really chemokines that are going to call in other cells. So we're going to be recruiting in other cells. It's not just that we made the blood vessel leaky, and so if they happen to come by, they can go in. we also are actively saying, hey guys, I need your help. Hey guys, please come here, with these. And in particular, neutrophils are going to be really important. And so we'll see a lot of the action of those neutrophils. This point that it's talking about with IL-12, all I really want you to know kind of about what it's talking about here is that these cytokines can also enhance adaptive immune responses or help make a good adaptive immune response. So they're sort of like saying like, hey, adaptive immune response, get yourself ready. And so yes, we can get other types of cells, sort of start to get other types of cells involved, including adaptive cells. And we can influence the metabolism, like what is shown here with IL-6. Immunometabolism is a pretty huge area that a lot of immunologists are spending a lot of time thinking about. And every time I read about it, I'm like, oh, that's amazing. And also, you're going to make me remember my biochem. Some of that metabolism that I thought I was never going to have to care about again. I suddenly have to care about again. But the idea is that some of these cytokines are going to influence metabolism of local fat and muscle cells. Some of that has some really interesting immunologic effects in terms of how well macrophages or neutrophils or whatever works. That's kind of stuff that we're just now learning about. But we also have learned that this is going to contribute to the heat in that area, and that potentially is going to and start to raise the temperature in that area. Why might it be beneficial to get heat in that area, to increase the temperature locally in that area? Yeah. To potentially kill the microbe. So if we raise the temperature, that might have some antimicrobial effects. And if you look at sort of very classic textbooks, they're always like, and we have heat, and it's all about killing the microbe. Now we've realized it also does some other stuff too. But the classic answer is that we kill the microbe with the heat. So you can see here are some of those effects on the blood vessel that I already showed you. I don't like the order of these slides at all, but I'll make it work. You can also see here is sort of the recruitment of the other cells happening with those chemokines. And so all of these things are sort of leading to that classic inflammatory response. There are a couple of other really important points I want to make about inflammatory cytokines and inflammation in general. One of them is that there are some other molecules that can look a little bit and like some of these inflammatory cytokines and chemokines. Guess what? You already learned about them. So when we talked about complement, I told you about C3A and C5A, these small products that are made as part of the complement cascade. Basically, C3A and C5A are acting like inflammatory cytokines and chemokines, and doing all the same stuff that inflammatory cytokines and chemokines are doing. So if you remember, we had these two little molecules that were being made. They bound to a receptor, and they have the exact same roles in terms of recruiting cells and in terms of influencing the blood vessel that our inflammatory cytokines and chemokines do. And so this is another place where you can see some of the stuff that I've been separating into like this box versus this box. Doesn't like to stay so separate. So what I've been telling you is largely about local inflammation. Those cytokines you can see are having effects on the blood vessel in that area, making changes to the blood vessel in that area, trying to get rid of the infection, particularly the bacterial infection in that area. But those cytokines can also leave that local area, and go through the blood into other parts of the body and have actions on other parts of the body as well. This happens in a bunch of situations. It might be particularly something that happens if you make a lot of the cytokines, but as you will see when I tell you what some of the things these effects are, you can imagine this happening in a fair number of inflammation types of situations. And so some of the effects of the inflammatory cytokines that are happening systemically or further away, not at the actual site, are shown here. One of them is that these cytokines, and you can see it's the cytokines I called out before. Those cytokines act on the liver. And when they act on the liver starts making a bunch of proteins. They're called the acute phase proteins. Guess what the acute phase proteins are? Basically, they're complement. And so the cytokines basically are signaling to the body, hey, there's something going on. Maybe we should make more of those complement proteins. They might be useful around now. And so we're suddenly going to get production of additional complement proteins. I had a doctor appointment, had some blood work done. And one of the things the doctor's like, well, we're going to look at your acute phase proteins, and tried to explain to me what they were. And I'm like, yeah, I got it. So that's something that is often measured when you go get blood work done. Those cytokines can also act on the bone marrow. They both tell the bone marrow, release whatever you got in terms of neutrophils, and also can spur more neutrophil production in the bone marrow. So again, okay, we're going to start using some of this stuff. We probably need more of it. Let's make some more. And let's release the SWAT team out of the bone marrow. Again, if you have blood work done and they say you have a high white count, it's mostly because you've just released a bunch of neutrophils. Probably because you've had some inflammation done. We can also see those cytokines influence the hypothalamus to influence body temperature and influence fat and muscle to also kind of mess with metabolism, which can have an effect on body temperature. The metabolism stuff, again, may have some other immunologic effects, though that's sort of newer stuff that we are learning about. And a lot of this does help with increasing body temperature to try to get rid of the microbe. So you can see this mentions increasing in body temperature. Sometimes I always talk about your mom or your grandma, but maybe your mom or your grandma is like a biomedical scientist and I shouldn't insult them. My mom taught first grade, so my mom is not a biomedical scientist. So we'll say, what would my mom call this increase in body temperature? What would she say happened here? Fever. And so those cytokines in one location of your body are going to go act on the brain, and that's going to induce fever. That's going to be good for some positive immunologic effects and to decrease microbial replication. Of course, if you had high body temperature for a long period of time, it would be really bad for your cells. Your cells don't like the high temperature either for a long time, but ideally what we want to do is if we stop the bacterial replication, keep their numbers down, our cells can survive it for sort of a short period of time. And so here you can basically see pictures of what I just told you in words. Look, those cytokines can act on the liver and make a bunch of proteins, particularly complement proteins, though we can also have some other proteins that are being made, like some proteins that are involved in blood clotting. Some of those cytokines can act on the bone marrow to tell the bone marrow, hey, bone marrow, release all the neutrophils. And we can also have some of these other metabolic effects I mentioned. And so if you make a lot of inflammatory cytokines and they're going through your body, you are going to have sort of a number of sort of body-wide effects, not just those local effects. And so again, in those larger quantities, you're going to have them entering circulation. They can lead to the acute phase proteins. They can lead to changes in blood cell production. They can also lead to things like sleepiness, lethargy, muscle pain, lack of appetite, nausea, that kind of blah feeling you feel when you feel sick. Is officially, the official word for that is malaise. All of those things can happen largely because you are making an innate immune inflammatory cytokine response. And you're kind of shifting resources towards immune defense away from other things, and getting your body in this state to be best for defense. So because I decided to do some stuff on Monday, that means I can also like go on like little interesting tangents more now. So one of the things I just mentioned, because I've had situations where students have decided that since this is the first they hear of cytokines, that all cytokines do inflammation. That is not the case. There are other kinds of cytokines that do all sorts of other things. So we are going to see other cytokines throughout this semester. But there are two kind of side-ish tangents that I can mention off of what's going on here. Sometimes people will have their sleepiness and muscle pain and blah, and maybe a little bit of fever. And we'll be like, oh my gosh, I'm sick, or oh my gosh, I have the flu. Sometimes people will in fact say that after they've gotten a vaccine, and they'll be like, the vaccine gave me the flu. If you notice, all of those symptoms actually have nothing to do with the microbe. There's no microbe hurting you doing this stuff. These are all things that your body is doing to help make a good immune response and to improve immune defense. A fever is something you did to yourself, not something that the microbe did to you. And so whenever one of my friends complains on social media about how bad they feel after a vaccine, I usually say something like, I'm so happy to hear your immune system is working as it should. None of them have punched me yet. But in fact, this is really kind of, the fact that these things are happening is really an indication of an immune response happening, not actually an indication that there's particular damage or something like that. Sometimes, I got some vaccines last Friday or last Saturday, and I was a little sleepy, and that probably meant that they were working. So like all of that's a good thing. So that's one thing to be aware of. There is some debate in the field about whether inhibiting these symptoms actually inhibits your immune response, and so whether you make, say, a bad adaptive response or a less good adaptive response if you take a fever reducer. People debate it very much in the field. There's not like clear evidence one way or another. I've seen evidence both ways. I do things like get my vaccine on a Saturday so that I can sleep on Sunday and not have to take anything, and then not risk it. But you do you. So that's sort of one general area of immunology that's really related to this. But I also want to mention another one just because I can. So you can think of lots of situations in your life where you have had inflammation. I'm asking this question in a weird way, sort of on purpose. If it ends up going too weird, then we'll forget it. David, you had any inflammation this week? Yeah? Why do you think I'm asking you specifically, and this week? Like, think about you and extracurricular activities I know about. Okay. Okay. So have you, maybe like at some point, like twisted an ankle or a knee or had, inflammation somewhere like that, right? So you can imagine you've all probably had the inflammation in that kind of situation as well, right? You can think about that kind of thing. So technically, this is what's happening. You had some cytokines were made, you changed the size of the blood vessel in your ankle, all of that, blah, 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 blah. But there's a question that comes up with all of this. But Brenna had a question first.
I don't know if it's here.
Maybe.
I was going to say, how then does the macrophages get...
So what's the question?
I was say, how do the macrophages get initiated when there's no...
Yeah, so if I just twist my ankle, how did the macrophage start making the cytokines? What did the macrophage sense to turn on those pattern recognition receptors and start making cytokines? This is another big area of research in the field. Officially, this is something called sterile inflammation. The idea is that it's inflammation, but there wasn't a microbe there. So how the heck does that happen? This is also somewhat controversial, but I think it's important to mention because if you think about it, we've all know this. It clearly happens because we can all think of situations where this happened to us. Pretty obvious. And what we think actually happens is that some of those pattern recognition receptors bind to not MAMPs or microbe-associated molecular patterns that I told you about before, but we have this idea that there's something called a DAMP or a damage-associated molecular pattern. So when some of your cells die or when some of your cells are damaged, they may release products that stimulate PRRs in macrophages. This is still very much, like I said, something that a lot of people are working on, but we think that there also may be ways that macrophages might sense products that are coming from other cell damage. And so maybe some of this is coming from bacteria, but maybe some of this is coming from the fact that there was a wound and damaged cells there. And so I just find that all particularly interesting because, again, it's something that's definitely happened to all of us. Yeah, Brian. Mm-hmm.
What are the benefits of inflammation to healing? Because everybody always says like ice it down, reduces swelling, like where did that respond?
Yeah, so generally, inflammation is and this is we're sort of at active research areas in the field. But I mentioned that there are some of those metabolic changes that I think we're realizing that may actually be beneficial. But we're also realizing how closely a lot of these inflammatory cytokines and sort of inflammation processes are linked to wound healing. And so you might get better healing. You might get better, say, scar formation or fibrosis formation or things like that. Because you have the inflammatory cytokine. And so the benefit, I think, is probably in the giving you a good area for healing. And that's something that a lot of immunologists haven't thought about as much and are starting to only think about more now. But I think it's more about that wound healing link, and the sort of how you deal with tissue damage. And how you kind of best repair a tissue from tissue damage is where that positive is going to come in. So that's my short answer on that. Other questions? All right. So at this point, we have seen our wound. We've seen our macrophage that lived in the tissue sense a microbe like a bacteria. We've seen that macrophage make some cytokines, and we've seen those cytokines have an effect. And one of the big effects that we have seen of those inflammatory cytokines is that we have called in some other cells to come help out in this region. And so we see these other cells coming in. And so the final piece to all of this is we have to think a little bit about these cells that are coming in and actually killing all the bacteria that may be in that location. The cell that is coming in this situation is really a neutrophil. And so they're not labeled as such here, but at this point, we're really looking at a neutrophil and its ability to get rid of those invading bacteria. I often use various insults for my neutrophils. I'm learning more and more about reasons why I shouldn't, but because I think about macrophages, which are the other innate immune phagocytic cell, sometimes I'm like, oh, neutrophils, those are the crappy ones. I work on the good ones. But no, I shouldn't insult the neutrophils. And so the neutrophils are going to come in and are going to get rid of invading bacteria. Again, sometimes I think of them as like the SWAT team that's going to come in and sort of clear up the area. And the neutrophils are largely working on this by phagocytosis. And so you can see that neutrophil actually stretching out little processes of its cell membrane and making a little organelle out of that process to internalize or sort of eat that bacteria. The overall cell biologic process here is called phagocytosis. And since we made this organelle in phagocytosis, we call it a phagosome. It's the phagocytosis made organelle. Yeah.
The phagosome is within the...
Yeah, so the phagosome would be this thing that's made after we've closed off and engulfed the microbe. And then what you can see is this is sort of, there are different ways that you can think about this, but eventually that phagosome is going to get more acidic. It may fuse with other compartments. It may also fuse with some of the granules that are in our granulocytes, which were full of toxic products. And then we're going to completely digest and kill that microbe. And so we're going to break up that microbe entirely. You can see that the version of this from your textbook here. So you can see here is our microbe. Here's the neutrophil engulfing it. Now that we have it inside, we would call this a phagosome. And we can have all of these other little granules that were full of all sorts of fun toxic stuff merge with the phagosome. And now we can completely destroy the microbe, bring it down to its individual component parts. You know, you could actually, in some cases, get it down to amino acids, things like that. I want to tell you on the next couple of slides about some of the specific toxic products. But before I do that, I think it's important to do one other thing in thinking about phagocytosis. Again, partially because I can, which is that I want to show you a neutrophil doing phagocytosis. This video is all over YouTube. This is like a famous video in immunology. I swear, sometimes I root for the bacteria just because I've seen it so many times. Literally, if you Google this video, or if you look it up on YouTube, you can find it set to any music you want. Like, you can find it set to, like, the Cops theme song, and, like, all sorts of funny things. We're not going to deal with that now, but if you're looking for it, you can find it. So in this video, we've got a neutrophil, which is this cell here. And we've got a bunch of bacteria. They're staph. They're little black dots, which will be a little easier to see once I hit play. We've also got these cells. Those are contaminating red blood cells. They aren't actually part of the experiment. It was just that the person happened to have red blood cells in their prep. Although they end up kind of being fun in the video because they look like little obstacles. And so we're going to watch this neutrophil both do some chemotaxis and do some phagocytosis. Yes, that's a neutrophil. That's still a red blood cell. That's still the bacteria. And go. Mm-hmm. It's pretty old. And so you can see that basically that neutrophil, if we watch back here, you're going to be able to see it change the shape of its membrane and actually come around that bacteria and fully internalize that bacteria, which is going to allow for that bacteria to be degraded. So that's the very famous immunology video. I'm not going to show you a German Shepherd puppy brawl. Sorry. I will also tell you that when I when I used to teach cell bio, I had there's a similar video that tries to show phagocytosis of where an amoeba does phagocytosis of a paramecium. But then someone in the class cried because they were sad for the paramecium, so I couldn't show that one anymore. So please don't feel too bad for the bacteria. Yes, David.
Now, is that video sped up?
It is sped up. I don't know the exact numbers, but it is sped up. So that is a much more fun version than looking at this as a static image. But once that microbe has been phagocytosed, it will be degraded, both because of the acidity of these compartments and some of the specific products in the granules. Remember, our neutrophils are granulocyte that are going to fuse with the phagosome. When I think specifically about the products that are in that phagosome, one of the things that I think about is that the neutrophil is actually making a bunch of enzymes so that it can make some chemical compounds, particularly that neutrophil in those granules will be making hydrogen peroxide. It will be making hypochlorous acid. It will usually be making nitric oxide, potentially a hydroxyl radical. All of those things are going to be being produced from oxygen and from nitrogen-containing compounds by the neutrophil. So the neutrophil is just going to have a store of these chemicals, and those are going to work to produce to kill the microbe. The overall process of making all of these compounds is called respiratory burst. If we're making the ones that contain oxygen, like hydrogen peroxide or like hypochlorous acid, we sometimes just bulk refer to those as the reactive oxygen species. When we're making the ones that have nitrogen in it, like nitric oxide, we sometimes talk about the reactive nitrogen species. And so the neutrophils are making a bunch of these in order to try to kill the microbe. How many of you have ever had a bottle or used a bottle of hydrogen peroxide for anything? Pretty much everybody. So hydrogen peroxide is really good at killing bacteria. You know that. You use it sometimes for things. And so basically the neutrophil just makes a ton of it to kill bacteria that are in it. You've also used hypochlorous acid, though you may not know it. Hypochlorous acid is the active ingredient in bleach. And so basically the neutrophil is making bleach and then bleaching the bacteria. So pretty straightforward way of killing those microbes. One of the things that also, and so you can see here are my toxic oxygen and nitrogen products. One of the things that will help get rid of those microbes is the fact that we are in a very acid environment. And so the acid actually is helpful. But we also have a whole bunch of antimicrobial peptides and various enzymes in some of these granules that are going to get fused with the phagosome and that are going to help destroy that microbe. So we're going to have a lot of the antimicrobial peptides I told you about before happen to be also found in neutrophils. So neutrophils can kill the bacteria. There also are things like lysozyme. There are enzymes to break down the proteins. There are enzymes to break down the lipids. There are enzymes to break down the carbohydrates. There are enzymes to break down the nucleic acids. Many of them happen to be versions of those enzymes that work really well in acid. And so basically we can completely break that microbe down into its component parts. So we've killed it, we've completely broken it down. And you can sort of imagine that as being really useful for, hey, look, now I got some new metabolic products. I got some carbon. I didn't even have to do any eating from killing this bacteria. There's one more step or one more kind of key thing that happens when we're thinking about this process with the neutrophil. If you looked back to day 2 of this class, when I showed you the table of cells of the immune system, there was one other thing that sort of was unique about neutrophils, or that came out about neutrophils. And that is that neutrophils are really, really short-lived. Neutrophils live for a really short time. They were the ones that sometimes only live for like hours. And so once the neutrophil has done this phagocytosis, the neutrophil is not going to live very long. And so the final thing that will happen is that neutrophil is going to die. Again, my favorites, the macrophages that I think are so much better, they don't die after they do phagocytosis. They do other stuff. But that's not right now's problem. The neutrophils, they die. And so usually what will happen is our neutrophil will phagocytose a bacteria, will break it down all the way, and then the neutrophil itself will die. And that neutrophil will get phagocytosed by a macrophage. So the macrophage will come and like clean up the body. And so neutrophils are, again, really short-lived. That can be pretty nice because if you can imagine, if the neutrophil couldn't kill the bacteria, well, then the neutrophil itself will just die and give us another chance to kill it in the macrophage. So the macrophage is really going to be the final thing that's going to get those metabolic products because the neutrophil is so short-lived. You, again, already knew about this because if you think about different cuts or wounds that you've had, you know how there's that white goopy stuff that sort of happens as you're healing? It's pus. The white goopy stuff, the pus, is just the dead neutrophils on the surface of your skin. That's all it is. I once upon a time did when I made this slide, I did a Google image search because I was going to show you pus. Don't ever do a Google image search for pus. You will see all sorts of things you did not want to see. So that's why it just says pus on that slide. The neutrophils, we have also learned, can do some other interesting things when they are undergoing death. And so I have to give them credit here. This is a cool thing about neutrophils. So neutrophils may not undergo just sort of your random apoptosis, everyday cell death. Neutrophils often seem to undergo a unique kind of cell death. And that cell death is known as NETosis. When the neutrophil dies, the neutrophil throws a bunch of its products that were inside of it outside of the cell. In particular, the products are the DNA and histones and sort of chromatin. And so basically the neutrophil is throwing a bunch of products outside of the cell. Those products are kind of sticky. And they end up forming an extracellular kind of trap. So if there are more bacteria around, they get stuck in the sticky trap. This is known as a neutrophil extracellular trap, sometimes referred to as a neutrophil net, because neutrophil extracellular trap, take the first letters and make it net. It always annoys me a little bit because we always say neutrophil nets, and I'm like, no, the N already stood for neutrophil. You don't have to say neutrophil twice. Okay. So what we're realizing now is that neutrophils do have this unique function in that they can throw out nets, when they are dying to sort of trap any other extracellular bacteria. Your textbook actually just shows you images of this. And so here you can see this is all sort of chromatin from neutrophils that have died, that has been extruded from the cell, that have this bacteria all stuck in it. And so it's this nice net. Here you can also see a bunch of bacteria that are stuck into a neutrophil net. And so when you think about neutrophils, yes, I often think of them as the SWAT team, and in my mind, they're kind of the dumb one. My macrophage does all sorts of fancier things. But neutrophils are also like Spider-Man, in that they're basically throwing out nets to catch any extracellular microbes. So this is also related to Brian's question a little bit earlier, which sort of gets to, the public perception that inflammation has. Usually when people think about inflammation, they think of it as a bad word. They think of inflammation as a problem that you got to get rid of. And what I want you to note here is that, no, you actually need inflammation to do host defense, to do tissue repair, and to kind of restore homeostasis. If you can't do inflammation, all those things are going to be bad, are not going to go well. But, as you might guess, there are also some downsides of inflammation. And so there is kind of the bad side of it. People often think about the bad side and don't think about the good side. I want you to remember, there's a good side. It would be really bad if you did no inflammation. If I just watched all of the random makeup TV commercials that are going to get rid of my inflammation, if they really 100% got rid of it, that would be very bad. The problem with inflammation is if inflammation happens for too long, so not for a really short period of time, but for a really long period of time, or if inflammation happens in too many places. So if you're having inflammation happening in many body locations, then that can be a problem. And so you can see that if inflammation goes wrong, then we start to get different kinds of autoimmunity or other kinds of disease states. We can sometimes see fibrosis or tumor growth, and we can see a number of other diseases. So there are a lot of diseases that are related to too much inflammation. If it's unregulated, too long, too much, wrong place. But you gotta have some. One of the most important things that ideas that immunologists spend time thinking about is we love Goldilocks in immunology. We talk about Goldilocks all the time. Because for most processes that we're going to talk about this semester, too much is a problem, too little is a problem, and what you really want to have is just right. Just like how Goldilocks wanted her porridge. And so inflammation's the same way. Too much inflammation, bad. Too little inflammation, bad. You want to have just the right sort of middle amount. So to just show you a couple of details related to inflammation being problematic, one of them is something called septic shock. Septic shock is a unfortunately common cause of death in a lot of hospital settings where people are going to the hospital with sort of bacterial infections. And the issue with septic shock is that with septic shock, the macrophage that starts out the whole process is not a macrophage in some special tissue like the skin. It's a macrophage that's like in the bloodstream. And so when we saw local inflammation before, we saw that we would change the vasculature in that one local site, like in your hand. And that would be okay, that would help you heal. If, however, those cytokines are being made throughout your entire body by macrophages in the blood, all of the blood vessels in your entire body are going to dilate. All of the blood vessels in your entire body are going to get leaky. And so you're not just going to have little red inflammation in the hand. you're going to have this massive inflammation body-wide. And this is sort of the very, very extreme example of sort of massive, quick inflammation going on body-wide. So what do you think happens if all of your blood vessels and your entire body dilate? Yeah. Why do you become Jabba the Hutt? Because the blood starts leaking out into all your tissues. So there's going to be a bunch of swelling. You can see it says systemic edema. That actually is fancy terms for a lot of swelling throughout your whole body. Okay? So you're going to have a lot of swelling. What else is going to happen? Yeah. Well, your heart's going to have some weird issues with output, but there's another thing that's sort of related to the leak, the Jabba the Hutt leakiness that we got to get to that sort of relates here. Yeah. Your temperature is probably going to shoot up. We still got one more thing. So all the blood leaks out of your blood vessels. Now what happens to your blood vessels? Oh. What happens to your blood vessels if all the blood leaks? There's no blood in the blood vessels, right? Because it all leaked. The blood vessels actually collapse because there's nothing in them. And so we get decreased blood volume because there's no more blood in the vessels. It's all everywhere else. We have no proteins in the blood. And we end up with collapse of the blood vessels. This is a problem for your heart, as you well mentioned here. And so we eventually get things like blood clotting in weird places throughout the body. We get organ failure of all sorts of organs, and this tends to lead to death of patients if this happens, and they tend to get a bloodstream infection. And so this is sort of the most prominent example of sort of what can happen with excess inflammation. The assignment that you guys have for next Friday involves some readings about some discoveries with PRRs and the Nobel Prize. And one of the reasons why people are so excited about about understanding PRRs is that if we could understand, what's going on, all the steps to producing the cytokines, we could stop the cytokines. And we could stop things like this, but also any other one of those diseases that may be related to too much inflammation. Yeah?
You might have said it and I missed it, but how rare is septic shock? Like, does it actually have a large amount of deaths?
Yes. It's a leading cause of death of people who are hospitalized. It's pretty big, yeah. So you try to not get to that point. But there are some other things that can happen with inflammation as well. So I guess, let's see. Now I'll start with this one. I told you about these cytokines influencing blood vessel permeability of the blood vessel wall. Now imagine that you don't necessarily have to have a huge amount of that cytokine being made. Like you're not in full-on sepsis. But let's imagine you make too much of that, just a little bit too much every day. All the blood vessels in your body are going to get messed with a little bit every day. And eventually you're probably going to end up with like problems with blood vessel permeability that might lead you to cardiovascular disease or might lead to clots like thrombus. The heart's a muscle. If it's getting cytokine hitting it all the time, remember we talked about muscles changing their sort of output because of metabolism, you're going to get changes to heart output, and you're going to, again, potentially get cardiovascular disease. If you get these cytokines constantly hitting other muscles and trying to constantly mess with the metabolism of those other muscles, you might end up with something like insulin resistance, which is the sort of thing that's leading to type 2 diabetes. And so too much of any of these cytokines, even if it's low level, but it's like you have it for low level for 20 years when you weren't supposed to, can have a lot of different effects. And so we have now been able to realize that chronic inflammation, so basically, even if it's kind of a lowish level, but you just have it going on for a long time, is going to lead to all sorts of tissue changes, angiogenesis, fibrosis, changes in insulin signaling, is very much related to obesity, leading to things like type 2 diabetes, heart disease, atherosclerosis, organ failure, cancer, Alzheimer's, autoimmune disease, autoinflammatory disease, allergies, transplant rejection. Basically, if you can think of it, I can relate it to chronic inflammation. And this is the reason why it was such a big deal that we started to understand this process of how the macrophage does sensing of a product and induces these cytokines. Because the more we can understand about that, we could potentially stop this chronic inflammation and come up with treatments that are impacting all of these disease states. It seems as though some of the reasons why chronic inflammation may occur might be due to things like you having a weird response to the microbes in your body, like commensals, or you having infections that you're not clearing for some reason. Some of that might relate to your genetics, or some of that might relate to other things. We are finding more and more ways that diet actually can influence inflammation. There are certain types of lipids, for example, that seem to signal to PRRs. And so that can lead to excess inflammation based on diet, but then a number of other kind of things in your life might trigger PRRs to kind of trigger background chronic inflammation. And so what you might notice from this is that suddenly you care a lot more about those PRRs and what might bind to them, if you want to control some of these things in your life. And happily, there are two areas I'm going to spend time on Monday. And one of them is individual PRRs and sort of details of how they work and what they bind. And so that's one area that we're going to deal with on Monday. The other area I want to deal with on Monday is a little bit of a compare and contrast with this process that I've told you about here. So I've told you about this very classic process of inflammation here. And if you look at this image shows a bacterial cell. Bacteria is being the pathogen. If the pathogen is instead a virus, some of the details of this process look different. And that's because viruses have some different biology than bacteria. They are largely, for example, replicating inside of cells, while this bacteria is outside of cells, among other things. And so the best classic response to a virus in this innate immune response is going to look a little different. And so we're going to start with thinking about kind of what's the virus version of this inflammation process. How does it work for viruses? And then we will talk about individual PRRs and some of the things that they bind. So I will see you guys on Monday.